Gene, we first met, I don't know, maybe 10 years ago when you were at Nestle and doing so much amazing work with the Nestle Health Sciences Institute on various trials on medical foods and nutraceuticals. And I remember looking forward to serving on that scientific advisory board just to hear about your work, which was the most exciting that was going on there. And we're so thrilled to have you now join us to carry out our clinical trials on products that are available right now for people that are safe, whether it's a safe drug or a natural product or a supplement. And so just want to say on behalf of the McCance Center, we're thrilled to have you and to get going with this new, exciting new era in the McCance Center for drug discovery and clinical trials around known safe drugs and combinations of known safe drugs and natural products. So welcome.
Thank you very much, Rudy. I'm very happy to be here. And I also enjoyed your presence in the Scientific Advisory Board when I was at Nestle. I also look forward to those moments, spending time. And that was an ambitious project that we were trying to do there in Switzerland. And I'm glad to be back here in Boston and moving things forward from a clinical trial standpoint.
Yeah, before we get into this exciting new program for drug trials at McCance Center that you're going to direct, and it's going to be a great collaboration with the Genetics and Aging Research Unit where we are now, that has come up with these various drugs and natural products that might be useful for repurposing in Alzheimer's. You've made your mark recently with a really landmark trial in terms of potential on omega-3s, the DHA and EPA, in a trial on Alzheimer's. Maybe spend a couple of minutes telling us about that.
Sure, be happy to. We, in 2012, we identified a combination of omega-3 fatty acids, EPA and DHA, that they appeared to be influencing certain parts of the brain. Not Alzheimer's type pathology, but more of the vascular components, what we call white matter lesions, small vessel, small disease in the brain. And we were able to secure funding for a 3-year trial, completely biomarker based. So we gave one group omega-3 active and one group without the omega-3s, a placebo. And we looked.
What was in the placebo?
It was soybean oil. So it's high in omega-6.
Okay. Oh, okay, so it still was an oil-based capsule.
So it was an oil-based capsule, matched on taste and smell.
Right.
And we followed people for 3 years, and we did neuroimaging annually, and we identified some interesting findings. We found that the white matter integrity of the brain appears to be affected by the omega-3 fatty acids. So those people that have demyelination in the brain over time, the omega-3s seem to prevent that. Now, these are people that don't have disease. These are people that have these white matter lesions and have suboptimum omega-3 fatty acid baseline. So
unpack that for people who are watching this who don't know what demyelination is. Like, what does that mean?
Yeah, so there are these myelin sheaths across the axon.
The insulation.
Yeah, they're insulating factors that
insulating
accelerate
the
axons
and dendrites. And
yeah, and so
like the plastic wire around your wires and your electronic components.
They help with conductance
Right.
and processing speed and executive functions, these cognitive processes. So the omega-3s appear to be influencing those parameters and those mechanisms much more than the Alzheimer's type pathology, at least in this particular study. The unique aspect of this study from a design standpoint and some of the things we want to do here at the McCance Center is that it's very targeted. It's a very targeted intervention in the sense that we enroll people that have suboptimum omega-3 status using an objective biomarker, a blood test. Plus, we do neuroimaging to make sure that folks have a particular pathology in their brain that we think is sensitive to the intervention.
But the omega-3s, I thought I saw you speak once at a seminar together we did in Tufts, where you were looking at tangles with omega-3s, the neurofibrillary tangles in Alzheimer's. Where did that end up going? Did that not?
Yeah,
because you had some potential effect there, right?
Well, so one of the omega-3s that we chose, DHA, does have effects on beta amyloid production. Right. And so the EPA, the one that we use that was very high in concentration, has more anti-inflammatory effects. It can lower soluble ICAM and some of these endothelial dysfunction, so it protects basically the small blood vessels of the brain, which may be an upstream event to what's happening in the central nervous system.
So what, if you think about dementia, right? Alzheimer's is 60 to 80%. I think Lewy body dementia is next at about 5 or 6%. But next is vascular dementia.
Right.
And vascular dementia is like dementia that's subsequent to a stroke, or just neurovascular issues like unhealthy heart, unhealthy brain, many strokes, many ischemic events. So are you, would you, and of course the vascular events can put you at higher risk for Alzheimer's
That's right.
type dementia. I just finished the fourth one is frontal lobe dementia, of course, which is mainly just tauopathy or tangles. So would you say omega-3 of those 4 dementias is mostly for neurovascular dementia and small vessel, but then will leak over to helping with Alzheimer's disease as well? I mean, how would you frame that?
Yeah, I think it depends on the omega-3 that's being used. So the EPA, I think, is having more vascular effects, and the DHA is having more central nervous system
Really?
effects.
So different effects.
So if you combine the two, then you may get the double effect. Of course, we have to show that.
Well, I take a vegan omega-3 every morning with DHA and EPA that comes from algae, because as a vegetarian,
algae. Yep.
I don't do fish oil.
Yep.
But
So our study did use fish oil. It was a
Right.
marine source.
yeah,
And,
but I think we should also say that with fish oil, you want to use clean fish oil, because if you're buying that cheap fish oil, like bucket O fish oil,
That's
that
true.
costs like $5 for a bucket, you're probably getting some mercury and heavy metals in there. So you got to get the nice, clean, filtered stuff, right?
That's
If you-
right.
Yeah.
Well, I think one aspect of the trial, something else we learned, is that those that actually get in the bloodstream a certain level of these omega-3s seem to get benefit. If you don't hit that threshold in the bloodstream, which is making it available through the blood-brain barrier into the central nervous system, you don't get the effects.
Oh, okay.
So two key things from the trial.
But wait, you're saying not all EPA and DHA that you take is going to get through, or
It's not. It's not. So we gave 1.65 grams, and there's only about half of the cohort actually met the threshold that were taking the pill. So there's something else going on. It could be genetic. We did see that those carrying the ApoE4 allele, those folks seem to benefit more. So there seems to be some gene-nutrient interaction. And again, I think this is another area that together we could do a lot of work together. This idea of personalized or precision medicine or precision nutrition.
In fact, that's what I was going to ask you about next, because we've been doing Alzheimer's genomics forever. I mean, I've been doing it since it started. I did the first Alzheimer's genetic linkage study 40 years ago.
Right.
I was very young. As literally a kid at the time here. And so we're developing this polygenic risk assessment. How do 100 different genes with hundreds of different variants in them combine to determine your overall risk, familial or genetic risk? And then you've come up with something called the nutrition index, which is more in the biomarker realm. And of course, we have biomarkers for Alzheimer's that we look at in the McCance Center, biomarkers in the blood for amyloid and the tangles and inflammation. And you're like backing it up a step and saying, whoa, wait, before we even get to that problem of having those pathologies in the brain, setting off Alzheimer's where you can see biomarkers in the blood, you're looking at a step back. How does your nutrition index of the food you're eating every day, put your brain at risk or protect your brain? So maybe spend a couple of minutes on that.
Yeah, no, I think there's several important things to talk about here. So I think this nutritional risk index is basically an objective indicator of somebody's diet, okay?
Yeah.
But it's not just their diet, it's actually what's making it into the bloodstream. So the food has gone through digestion, absorption, assimilation, so it's made it into its active form into the bloodstream and readily available for transport into the brain. Just because somebody's eating well, it doesn't mean that you actually have that nourishing liquor the brain needs in the bloodstream. So some of the genetic factors that would influence, for example, omega-3 fatty acids, the FADS genes, and some of these that folks have an inability to effectively, as they age, convert the essential fatty acids, alpha-linoleic acid, into these longer polyunsaturated fatty acids that are abundant in the brain. So these factors are important. The other key aspect is when you put together these polygenic risk scores, which would be fantastic to have clinically to understand people's genetic risk. I think a big open question for the field is how would that relate to nutritional status? So for example, if I had optimum nutritional status and I have genetic risk, will it mute the genetic risk or will it not?
So if we had 1,000 subjects, like in a McCance Center project, of the many we're going to do with you, right? If we had 1,000 subjects and we determined their nutrition index in their blood, and you use that like a trait, like red hair, right? And then you do whole genome sequencing on those people, and you say, okay, when in this person with whole genome sequencing, I would find 60 million spots in your 3 billion bases of DNA that are different than mine. Okay, then I could ask, which of those 60 million variants that are different between you and me or different in the population have an influence on your nutritional index? Because then you can start to merge what you're eating in terms of diet together with your genetics to say, how does that merge together to determine your own nutritional index? And then that's going to directly translate to your brain health.
That's right.
Right?
That's the whole notion of precision medicine. I mean, I think that's where we need to go.
So that's a project we will do in the McCance Center based on the work you're bringing with you. So maybe, we can, and what I like about the nutritional index is it's early prevention, right? The whole mantra of the McCance Center is to be proactive, right? I always like to say the elephant in the room is that we don't diagnose neurodegenerative disease until the brain has deteriorated to the point of dysfunction. If Alzheimer's disease was diabetes, we would not diagnose diabetes until you lost half of the beta cells in your pancreas. It could no longer process glucose, insulin. Then, of course we don't do that. We diagnose diabetes while the pancreas is still healthy, but you're seeing problems with insulin and glucose and you start treating that. We don't do that for Alzheimer's or Parkinson's or other neurodegenerative disorders or neurological disorders. That's what we want to change. So I think, just to finish this part of the conversation, if we can start with the most basic things, nutritional index, how much are you sleeping, which is when the brain clears away that debris like amyloid that causes Alzheimer's. How much are you exercising? What is your stress level, right? I mean, stress itself induces inflammation in the brain. So at the McCance Center, we have the Brain Care Score, which takes into account all these lifestyle factors from diet to blood pressure to stress to wellness to social interaction, all on the lines of, what I encapsulated as SHIELD. SHIELD, your brain is sleep, 7 to 8 hours. H, handle stress. I, stay interactive with others no matter how old you get, stay social, it's good for the brain. E, exercise. I try to do 30 minutes on the recumbent bike every day. But even a brisk walk for an hour is fine. Get that blood flow going. L, learn new things, which hopefully people are doing while they're listening to us right now, unless we're putting them to sleep or making them hungry, and that might still help if they have a healthy snack. And diet, right? So diet is plant-based and Mediterranean diet. Less processed foods, junk foods, less sugar, less fat, salt, less processed stuff, right? So if you go through all of that we can guide patients coming to us on SHIELD and how to stay healthy and how to practice early intervention and lifestyle to prevent brain disease. But beyond preventing brain disease, we want to think about how to create, no matter what your age is, at the McCance Center, how to create brain and mind Olympians, champions. In other words, how do we preserve and promote brain health, right? That's what you offer with what you're doing on this nutritional side, is having people have just basically better functioning brains and more vital minds, clearer mentality, and which is a whole nother level back from saying let's wait till the brain is sick and then try to fix it after it's already gotten to the point of cognitive function issues or movement issues. But this is really central to McCance, is preserve and promote brain health and prevent brain disease before it gets too far. So the other way we can do that is early detection, early intervention of the most common brain diseases. And of course, Alzheimer's is the one that we think about the most. It's most cases of dementia are Alzheimer's disease. So I'll just spend 2 minutes telling you about what we've done in the last 8 years. Is we created Alzheimer's in a dish. So this is basically a mini human brain organoid, gel like the brain is jello, right? Not jello, but gel-like material, 3 pounds of gel in your brain. And we create little paper punch-sized bits of gel that have in it the different human cells of the brain interacting, neurons, glia, et cetera, et cetera. And then we introduce into those cells the genes that cause Alzheimer's. So they create the formation of the amyloid plaque and the tangles that kill the neurons, and most importantly, the neuroinflammation, which is induced by the plaques and tangles, which kills 10 times more neurons. So we basically can create in a dish in 30 days in a paper punch-sized mini human brain organoid what takes 30 years to happen in a human. So in Alzheimer's disease, the pathology begins up to 30 years before symptoms. The plaque is starting to form. And then it takes 30 years before you get plaque to tangle, spreading tangles, and then finally enough inflammation in the brain to cause the loss of neurons and synapses and leading to dementia. So using that model, if we can see all that in 30 days, this has made drug screening and screening of natural products 100 times faster, 100 times cheaper. And so we've been doing it. This is what we've been doing. And now we've identified well over 200 known drugs that are considered safe because they're approved, and natural products that are generally regarded as safe by the FDA. And we're looking at combinations of these to target specific Alzheimer's pathologies, but the key is to use these early. We want to start looking for biomarkers in the blood. Who's starting to form amyloid, even though they don't have any signs of dementia? Who's starting to form tangles? Who's starting to get inflammation?
High risk.
And higher risk. And then start treating them with combinations of these compounds in clinical trials. So that's why we're so excited you're here, because you're going to run these clinical trials. So what do you just the feelings on that?
I mean, I think it's exciting. I think it's a big breakthrough, in the field, that work. And I personally, I've taken a different approach in identifying combinations from a population kind of top-down approach. And we have used algorithms or data-driven approaches to identify combinations that seem to have synergistic effects on different outcomes, brain health outcomes, whether it's white matter lesions we talked about earlier, whether it's total brain atrophy or some cognitive test. So it would be really interesting to now couple these two technologies together and come out the other end and see if we have some consistency. And if we do, I think it's a much more compelling, because I guess the top-down approach, what it gives you is it gives you concentration.
Can you dive in on that? What do you mean by that, top-down?
Well, I mean, looking at population levels of
Right.
circulating nutrients and bioactives in the bloodstream.
Right.
And so you basically do metabolomics or something
Right.
along those lines, some kind of omics.
But once you get your result, how do you translate that to a drug or natural product you need?
Well, so the algorithm will give you a list of nutrients or bioactives that in combination as an entity, so you can scale an individual on, for example, 7 to 10 nutrients in the bloodstream, okay? And if you have certain levels of those particular
It's
nutrients.
like vitamins and...
Yeah, like a pattern, for
okay.
example. But it doesn't matter, it could be whatever you want it to be. So for example, you could put somebody on a particular type of diet and then look in the bloodstream of certain bioactives and apply this algorithm, and it's still going to give you combinations that it looks like some of these bioactives are working synergistically. And I think if we could break down that into your models, picking one or two, improving it in the in vitro model,
Right.
it would be a really nice proof to then come out the other ends. Because what it does from the top down, coming back to that definition, is that the nice part about that is that you understand the concentrations that appear neuroprotective or detrimental.
Yeah.
And so when you're designing a trial, you understand where the thresholds are you need to get into this zone of neuroprotection.
Yeah,
And that's
this is
another important aspect.
So exciting because we're bottom up, right? So we're taking, an index card sized tray with 96 holes in it, or wells.
Right.
Each one has a mini human brain organoid that over 30 days first makes amyloid plaque, just like somebody might make when they're 50 years old. And then those plaques start causing tangles to form, which start to kill the nerve cells. And then toward the end, the glial cells get upset by this, and you get, just like in COVID, you get a cytokine storm. But in this case, it's not COVID doing it in your lungs or your blood vessels. It's the plaques and tangles are causing it in the brain. And then that cytokine storm causes inflammation. And in all these diseases, it's always inflammation is the end point that does the most damage.
Right.
That finally leads to symptoms. Even in COVID itself doesn't make you that sick or kill you. It causes inflammation.
Irresolved
Right?
inflammation.
That irresolved inflammation. So we have now, the drugs and natural products that took us the last 8 years. Which ones stop the plaques? Which ones prevent the tangles? Which ones tamp down the inflammation? And some of these natural products are going to directly relate to the bioactives and nutrients that you're looking at in your in large populations, looking in their blood. So when we merge all that, we can make new combinations based on the top down and the bottom up. But the real power is the fact that now we can do rapid trials, right? So people hear clinical trials and are like, oh yeah, they all fail, right? I mean, clinical trials from big pharma cost hundreds of millions of dollars, and they finally come out after all that money and time and they say, it failed, right? I mean, recently we had a success in Alzheimer's with lecanemab from Eisai with Biogen, which is an amyloid antibody, where they got small but significant clinical benefit. We're developing a drug that hits amyloid because the immunotherapy is expensive and it's not particularly safe. We're developing a little white pill that we hope will do the same thing. That's just going into a new drug that's going into phase 1 trials with the McCance Center next year. But in these clinical trials, they're cumbersome and they're not, and the way they're done, the odds are more likely for failure, right? So we're changing that by looking at biomarkers. So in other words, we'll take one of your top-down natural products or drugs implicated with one of the things that came out of our dish model, combine that. But we can do small trials where each person serves as their own placebo. That means that, and what you're looking at is blood-based biomarkers. So you're asking, are there changes in specific biomarkers in the blood of the subject that suggest the drug is working in them, hitting the pathology it should? So for example, if you have a drug targeting amyloid or tangles, or this one's targeting inflammation, you look at blood-based biomarkers for that pathology. These trials can be done in 6 months. They can be done at a fraction, a sliver of the cost of the big pharmaceutical trials. And all you're asking is this combination that I'm predicting should stop plaques and tangles or inflammation working?
Right.
And if that's the case, then we can
Confirmation
call up
in
Big
a sense.
Pharma and say, okay, we have a combination that actually we show through biomarkers that we have engagement of the target. We show that we're stopping plaques and tangles. Now we want to see if a big trial with that combination is going to cost a lot of money is going to lead to clinical benefit, make somebody cognitively better.
Robust
That trial is
proof
going
of
to
concept.
That trial is going to be expensive. But our purpose at McCance and the work we're going to do together is to de-risk these combinations so that the big money from big pharma, or from maybe a venture capital firm starts a new company.
Food and beverage companies.
Yeah, they'll throw the money in and say, oh, you've already shown it's safe. You've shown that it's engaging the right pathological target, and the biomarkers said that it's doing its job. Now let's see if it leads to clinical benefit. That's beyond us. That's going to cost hundreds of millions of dollars, but we can provide that pipeline now. That's not otherwise there, because pharma companies are not doing what we're doing. They're not testing supplements and natural products. They're not testing other people's drugs that are already known and safe.
That's right.
We're doing combinations of those that now become their own drug. And then we can de-risk that and say, hey, this looks pretty good. Work on this. And we become a pipeline for Big Pharma that goes beyond, the things they do, which usually are very similar across companies. And I think an example of this is the company Amylyx, where, 8 years ago, 2 kids from Brown University, I call them kids, one was a senior, one just graduated, Justin Klee and Josh Cohen, came here with an idea about a project to address neurodegeneration, address Alzheimer's disease by protecting the nerve cells from dying. We wanted to build a bulletproof vest for the nerve cells. And their dorm room idea was you have to have 2 sides of that vest, not just one. Like, up to that point, people were either protecting nerve cells based on the mitochondria or based on other organelles in the cell, trying to protect them. And the idea they came with was you got to do both. And so we did a project and found, tested 2 compounds, 2 sides of the bulletproof vest. And the preclinical work in the nerve cells in the dish were tremendous. And the next thing you know, they decided to start a company, Amylyx, which, for transparency, I have co-founding shares in that company. Not as much as they do, but I have to disclose that. But they went on then to start this company and do an ALS trial that worked, and now their ALS drug, this combination of one known drug and one known natural product, that combination, now called Relivrio here and Albrioza in Canada, was approved. So this shows what you can do with known drugs, known natural products in new combinations. And now this is the first new ALS drug that actually hits symptoms and extends lifespan that came from an idea from 2 kids in a dorm room using known entities that are already safe rather than having to create brand new drugs.
Right.
So we want to do that on steroids.
Shortens the timeline.
Yeah, we want to so what we've done is just set up this dish model. We've come up with 200 different compounds that hit these pathologies with age that threaten healthy aging and Alzheimer's. And now you're here to do those clinical trials, and we want to do as many as we can. We want to take as many shots on goal as we can.
Well, I'm excited to be here. I think, this idea of the different trial designs and where the field is focused, I think you brought up the Amylyx trial for ALS, and of course, these are people with the disease, right?
Right.
And the Alzheimer's field, most of the trials are focused in people with the disease. We call this tertiary prevention. But another aspect, is this more secondary prevention, which is the omega-3 trial. These are people that don't have the disease, but they have underlying pathology that looks like they're headed that way.
Yeah, that's our target.
And then the primary prevention, where you have maybe a genetic risk factor or family history or something along those lines, but you have no signs of pathology at all. So I think also filling up those 2 buckets with this proof of concept pipeline that can be taken on to the next kind of phase 3 style. But I think it's important to understand these different stages because it's also going to require different treatments. So for example, when you're combining medicines or natural bioactives, there's also a possibility that you get a stronger effect. This magnitude of effect can be maximized in some sense.
Yeah.
Certainly there's cases of that in nutrition, for example, but maybe also in the repurpose drug route as well, if you're touching on different mechanisms.
Yeah, and the thing is that with these repurposed drugs and repurposed natural products, when we do these combinations, we can do each trial for about $1 million versus a large phase 3 trial in big pharma can cost up to $1 billion.
Yeah,
So
it's a big difference.
1,000 times less. So we can our goal would be to do at least 5 to 10 of these combinations that look promising, $1 million each, run each trial, run biomarkers on the blood. We have a whole omics biomarkers core that we have, established at McCance Center, and then say, okay, are we doing our job? Are these two combinations that worked in a little mini, piece of gel in a dish, a mini human brain in a dish, real brain, I shouldn't say it's not a human brain in a dish, it's just gel with brain cells in it that recreate pathology. I don't want to get the idea that this is an actual mini human brain organoid in a dish. But sometimes we use those terms interchangeably. But the fact is that just because it worked on some gel in a dish with brain cells and to stop pathology, will this do it in a human? Will it get into the brain? Will it be safe enough? Will it be efficacious enough? And the thing is, because we have biomarkers now that act as surrogates, markers for this pathology, how much amyloid is there? Are we bringing it down? How many tangles are there in the brain? Are we bringing those down? Are we reducing inflammation? Right? These are the trials we'll do. And if we get, for the combinations that best hit this pathology based on the biomarkers, then we hope to be a pipeline for bigger efforts, whether they're in big pharma or new companies that get launched by others, or talk to VC firms who launch companies and say, look, you got to take this now and raise the big dollars that we don't have, 10, 20, 30, $100 million at a time to do the big trial and say, can we go from the smaller trial that said the drug worked to actual clinical benefit? And that clinical benefit, like you said, could be prevention, but it could also be treatment of people who are already suffering with disease.
That's right. Yeah, these larger primary prevention, secondary prevention trials are expensive. But
all the big trials, where you look for clinical benefit, are expensive.
Yeah, to look at clinical benefit. But the idea of combining different medicines and nutrients in these things, if we do get a more robust magnitude of effect, that also can have profound effects on the design of the trial. You don't need to study as many people and for as long.
Like a dozen versus 1,200.
That's right.
Just to look for proof of concept.
To see a signal concept, and then you have your pipeline proof of concept and ready to move on to the next stage.
But I want to emphasize for those who are listening to us right now that this is a new paradigm, right? It's rather than just come up with one drug and say, okay, let's spend a ton of money on the phase 2 and the phase 3 and see if it works clinically. Let's first see if it works on the pathology that causes the disease. See, this is different, right? We're saying, let's just see if it hits that pathology and use simple biomarkers in the blood to ask if we're reducing that pathology. And that says this combination has hope. It hit the right pathology as predicted from the dish model or from your top-down nutritional model. And now we can branch this out and find collaborators in the pharmaceutical industry to do the bigger trials. But we de-risked it, and we showed that it's worth betting on.
Yeah, we will answer the question of who's responding, and that's a very important question to understand moving to the next stage. And I think, like you said, these phase 2 trials, this kind of proof of concept, we're going to learn a lot.
Now, the business savvy people who are listening are going to say, you think Big Pharma is going to take 2 natural products I can buy on Amazon and invest in that? And that's a good question, right?
Yeah.
That's where this Amylyx model comes in. The Amylyx guys, the 2 kids, I still call them the kids, I can't help it. They were Sigma Chi fraternity members at Brown. I was a Sigma Chi in college. That's how they found me, actually. But that model says if you take 2 known entities, but you combine them, you can generate new intellectual property. Now, at McCance Center, we don't care about intellectual property. We're not for profit. But we have to think about it. Because after you do the first trial, the only way you're going to get Big Pharma to do the bigger trials, should you have gotten positive results, is to say that they can make money on it.
You have a protected asset.
No, big companies aren't going to test things they can't make money on that anybody could buy at the the drugstore. So, but you can combine known entities into a new drug like Amylyx did, and now you can offer not just the trial results showing that they work, but then there's intellectual property they can license that says if it works, they're for profit, they're pharma companies, that they can make money. And I know that people don't usually want to talk about money when they talk about research, but we have to think about sustainability. If we're going to take this big step to say, let's take what's already out there, known and safe drugs, and natural products as a treasure trove, And we've already determined which ones work best on all these different stages of Alzheimer's pathology. We've done that for the last 8 years with the Alzheimer's in a Dish model that Cure Alzheimer's Fund, funded from the beginning, and they funded the drug discovery. And none of this would have been possible without Cure Alzheimer's Fund, right? Then if you're going to do that, you have to think about, after you do your first trial, is it going to go anywhere, right? You might do a trial for $1 million and say, wow, this combination of two things I could buy on Amazon worked, right? But, if you want to turn it into something that's, an actual drug where you have, what to buy. If someone goes on Amazon and you say this supplement is good, there's 15 different versions. And half of them might not even be what you think it is.
Rudy, I think that's one of the
so
beauties of your model.
It has to be standardized.
That's one of the beauties of your model, is that you're identifying combinations that from a biology standpoint, we wouldn't necessarily be putting together. You
Exactly.
kind of have this data-driven approach
Yeah.
that has these novel combinations, and that's
Right.
an important part of the IP strategy. So I think there's a lot of opportunity.
But to go from our trials to a drug, it needs to be well controlled, standardized. You can't have the wild west where, you just buy something and hope it works. So that's why we want to come up with exactly the right formulations, even though they're known drugs or known natural products, the right combinations that are standardized, that then we can hand off. We hand them off to pharma and say, okay, now you spend the big bucks.
Right.
And guess what? It's worth it because we showed it works.
Right.
The McCance Center, our nonprofit entity, we did this, we showed it works, and here you go. And if they say, well, how are we going to make money on this? You say, there's intellectual property on it because it's a combination. We do that for sustainability, not for profit, but for sustainability. So Big Pharma will take it on, and the therapy will have legs after we're done with it. So I just want folks to understand that this is a whole new paradigm. It's a whole new model for drug discovery, never done before, that we're just pioneering now at the McCance Center. And that's why you're here.
Well, I'm excited to be here. I'm looking forward to it. Let's go.
Yeah.
Let's get going.
Let's go.